Okay, welcome to my 14th video from my 30-day challenge. I've chosen this day to celebrate because I didn't think I was going to make it here. 14, because that means it's 2 weeks straight that I've been making videos every day for your enjoyment, for your education, and for my legacy, to record for everyone my theory and my take on my theory. You'll get it straight out of my mouth. Anyway, today's video we'll be discussing what's the next step in the evolution after life initiated from encoding the first genetic information, which was the subject of last video. The genetic information Hi, my name's Eduardo Hernandez. I am the creator of Emergent Chemical Evolution, the newest theory on the origin of life. I'm also the author of the book by the same title. You can find the book in all major online retailers. Today's video, we'll be discussing what's the next step after life encoded the first information in its genetic information, initial genetic molecule, which would have been a strip of RNA. From the previous video, we determined that in order for life to begin evolution, it needed to have a basic system of genetic translation in place, which would have easily come about from viroids being reduced into transfer RNA, and also other strains of viroids being replicated with multiplication errors to become ribosomes. So there you have two components, the central components of genetic translation being initiated by viroids, the random chains of RNA. And since they originate from viroids, the transfer RNAs being a reduced structure of the viroid, and the ribosomes being a multiplied structure of that viroid, it would make it more easily for them to marry, to be able to have a conformation that would bind. So that solves a lot of issues, a lot of problems in how such a system would come about. Now you would have multiple strains of viroids replicating. Some would be, of course, more would contain more of the information for transfer RNA, and other strains of those viroids would have gathered information for ribosomes. So you have multiple strains of viroids with different genetic information, but again, available in an environment that's conducive to their survival and their multiplication. And like I said previously, with the ocean being one large cytoplasm, they would have bumped into each other regularly, therefore being able to interact throughout the whole time of the initiation of life for millions of years. They'd have been jostling and bumping into each other in the cytoplasm of the ocean. I mean, it probably didn't take millions of years because, as we know, viroids and RNA replicate very quickly. So any initiating replication would occur spontaneously all the time. So it probably wouldn't have taken millions of years to get it to a point where a regular system of translation would have been in place. That being said, multiple strains. So you have viroids replicating that would produce more readily transfer RNA, and you have other viroids that will more readily produce ribosomes, and you will have, therefore, different strains of that viroid. Therefore, you would create different lineages. This is important because they're looking for the universal common, the last universal common, last universal common ancestor, Luca. So they're looking for one, and in the cellular form, I claim by viroids being the first living molecule, albeit not self-replicating because it's a system that emerged thanks to the chaos engine at the initiation of life, according to my theory. But viroids is the last common viral ancestor, Luva. So it's a girl, not a boy. Luva is what my theory claims is the last common ancestor, is the last universal viral ancestor, because that's where life initiated. Starting with viroids solves so many issues. First of all, it solves the common genetic structure of life: viral, organic, cellular, fungal, all those different organisms, albeit have different coatings of membranes, of encapsulations, they have the same genetic information or genetic translation system. So that having it be Luva, the last universal viral ancestor from an informationless viroid, makes sense. It solves all these issues. How can organisms with different incompatible membrane structures, encapsulation structures, have the same genetic information if the chemistry and the biological processes are so different? This solves that issue. Why are they so different but yet have the same genetic information? Because that common lineage came from the viral stage, the viroid stage. So you'll have multiple viroids replicating. Some would be more readily cleave themselves, which is the normal catalytic function of RNA. They would normally cleave themselves to become transfer RNA, and other ones would multiply and become larger, gathering more information and gathering multiple copies of itself to become ribosomes. Therefore, you can have different lineages of viroids because you have different strains of the viroids that do different things. Now, these are things that are necessary for life, and they would eventually accumulate enough information to propagate that system. You can eventually have viroids become long enough to save information for protein translation or protein production. So you'll have strains now that either start with transfer RNA or start with ribosomes, but will further grow larger and collect what could be genetic information for the translation into proteins. So you have protein components that can be added to the length of the viroid making that a particular type of virus. Because no longer it will be a viroid. Remember, a viroid is a certain length and does not have protein information, has no translation information. So here you have a system beginning from non-information containing RNA molecule, which is the viroid, gaining information by making itself more susceptible to cleavage and becoming transfer RNA in one strain. Then you have another strain taking a conformation that makes it easier for it to gain more genetic information and become ribosomes. Either one can be independent and further gain on their own, gain more genetic information by having replication errors where their genetic information is multiplied. You have copies added. You'll have double the amount of the same information with each error occurring in replication. So you can have these strains grow larger over time. Of course, these starting with transfer RNAs and becoming larger, and these, the other ones with ribosomes and becoming larger. And then these eventually could have interacted with other strains of ribosomes and combined, therefore creating a larger RNA strain that has both ribosomal information and transfer RNA information, and becoming another lineage of that replicating RNA. That replicating RNA becomes its own strain, all of them, and they can go in different directions. Of course, you have a system of genetic translation in place now, and you have longer and longer strains, strips of RNA being multiplied, replicated by molecular polypeptides already available in the environment, in the ocean, that is acting like a cytoplasm at this point. Therefore, you will have more and more of these genetic information being available in the environment. Now, here's the thing. Again, like I said, eventually you will have these strains continue to grow and continue to carry more genetic material. Therefore, more material to be translated in the system. Therefore, easily these strains, as they become larger, can have strips of their RNA translated into polypeptides. Some of these will not just be replicated, some of them will be translated into proteins. And depending on the proteins translated on each strip of RNA, how that protein would garner that strip of RNA evolutionary advantage. Would that protein stabilize the RNA when it's not being translated, when it's in its normal state, let's say? It's when it's in its inactive stage. If that protein would attach to that same RNA and garner it some robustness, adding some stability to it and allowing that strip of RNA to survive better than other RNAs that don't have that protein, or another strain that creates a protein that aids in that RNA strip's replication, makes that replication faster, easier. So there's different ways that those additional protein information in the RNA could assist those RNA strips. Eventually, some RNA strips will produce proteins that could encapsulate the RNA. And you would get what I can only think of as the ancestor of the TMV. The tobacco mosaic virus is the simplest virus we know of. It only has 4 genes. It translates into 4 proteins, 2 of which encapsulate it, and the other 2, I believe, assist in its replication. And once that, hold on, there's a jet from the local airbase. Again, what was being I think I should, my voice will overpower now what's left. Again, all these components are going to be trapped in the currents of the chaos engine. They will be constantly transported through pH gradients. And from the article I read about TMV, from the original authors of the study on showing that viruses have genetic information and showing the replication of how that works, they said the most important factor in the assembly of and disassembly of these viruses in order for them to replicate is the pH of their environment, of the solution they find themselves in. So the pH has to be a certain pH for them to unravel, come apart, and replicate. And then the pH has to go back to another pH for the protein structures to become rigid and then interlocked with each other and encapsulate the RNA strip. So I can see that that could be one of the systems that allowed the propagation of certain strains of RNA. In that strain, it would be hard for it to further evolve into a larger organism. That's why I theorize that TMV is a very ancient virus that would have come about in during the close to the origin of life, and therefore find itself in niches that would have allowed it for its further existence as life changed the environment and the earth changed. So that being said about that strain, then you'll have other strains, which are other types of viruses that would garner information to create proteins that would encapsulate the genome. Now it will be considered a genome, the long strip of RNA, in multiple capsids, in multiple different types of protein encapsulation of the RNA information, so that you'll have different types of protein coats, which could become considered a type of membrane, a protective coat for the genetic information. And over time, you'll have different types of encapsulation. Icosahedral, rod shape like the tobacco mosaic virus, and amorphous, which would be proteins that don't have any regular structure, but that could still encapsulate and further evolve into a more complex type of encapsulation with eventually, when these viruses become large enough and contain enough information to do other processes themselves, which would make it easier for this virus to survive if this virus was able to produce itself a replication system within itself, so that viral strain becomes more complex. That virus would gather more information to produce proteins that would carry out other biological functions, eventually becoming a more complex cellular organism. There will be a stage where all the RNA strips available in the environment, not all, because viroids still exist, so they will continue to exist in the environment. There'll be a point where the virus will be the predominant organism in the environment. And as shown by the discovery of Mimi virus, which is a gigantic virus that can be seen in the microscope, that has millions of genes, well, millions of nucleic bases that produce proteins proteins that carry out metabolic processes that are not the same type of proteins seen in cellular organisms. They're different. They carry out the same functions as process fatty acids and process amino acids, but the information contained in those proteins are unlike anything seen in cellular life. So they're unique to that virus. And that fits with my theory that the viruses became more complex. And according to their niche and their environment, like as evolution states, they would either go extinct, stay the same, adapt to that environment, or evolve and change to live in other niches and other environments, which I theorize is how viruses fit between molecules to cellular life. The complexity would have been added as these viruses got larger. These viruses would have come up, being that they're fantastic agents of change, evolutionary agents, because they would replicate so quickly and get so many errors so quickly that their evolution would be on overdrive, turbo. They would have been able to come up with different types of encapsulation proteins, different type of proteins to do different biological functions within themselves, to different types of replication mechanisms, therefore becoming different types of lineages. So you would have certain viruses that would become animal cells. Other viruses will become plant cells. Other viruses will become fungal cells. Other viruses will become archaea. Because the chemistry that would have evolved would have nothing to do with each other. So one virus would create a type of encapsulation that would lead to a membrane that is like the animal membrane. Another type of virus would create a encapsulation system that would eventually evolve into plant cells with rigid cell walls. You would have other lineages that would lead to fungal type of processes that allow for a flexible, malleable type of membrane, allowing it to go through its different stages of life. And you have then the archaea, which are different types of organisms with very different types of membranes that have very different types of architecture and chemical composition that are incompatible with the other areas of life. So that's how you would get these different lineages and information encoded. That would have been the next step after the information to encode a genetic translation system was instilled in these strips of RNA. All right, I think I've proven my point in that point. The next video will further take this stage of life and point how each strain, well, I'll tell you what, we'll address each type of cellular kingdom of how they could evolve from viruses into that particular kingdom. Thank you very much. Tell your friends, subscribe, and have a good day.